So we were discussing the small intestine till duodenum. I mean the first part of the small intestine was done. In this we are going to discuss the second part, which is jejunum. Okay. So jejunum, first thing is, if we talk about the length of that, it's around 2.4 meter. This 2.4 meter, and these, after duodenum, the food enter into jejunum part, the middle part. This is the smallest than all. The second part and very smallest in these three parts. And so one more thing, when food enter into these, here a lot of enzyme will work. So that complete digestion would be done. So few enzymes which you will see, like its secret maltase. These are the enzyme maltase. This very basic concept, wherever you see the word A's, this means that have been enzyme. So maltase, if it's secreted, so this means what it will do, it will help in the breakdown of maltose. ASE word tells about that's an enzyme, but maltose is the macromolecule. Okay. Maltose is made up of what? That's made up of glucose plus glucose are known as maltose. Okay. We have a lot of other enzymes. Let me discuss all of that. So this is the first. Second enzyme which you will see here is that sucrase. This means it will break down what? It will break down sucrose. And sucrose, what? Sucrose is glucose plus fructose from the sucrose. Glucose plus fructose. This should be clear then. We move a slide little down. So the third you will see would be lactase. This means lactase will help in breakdown of lactose. And lactose is made up of glucose plus galactose. Glucose we do write like this, and galactose Ga. Okay. Then the fourth one, which is the last one here. That is, I think we really don't have that. And yes, peptidase that digest peptides. Let me just write it. That's also an enzyme, peptidase. Which help in breakdown of peptide bonds, and it's like, if we also call them as a nucleotide. When nucleotide are break down into what? They break down into, the enzyme which uses peptidase, but nucleotide is break down into nucleoside. This T break down into nucleoside. This means, if we talk about the difference between both of these, in nucleoside there is removal of phosphate, but in nucleotide there is addition. Sugar, ribose sugar, phosphate and base is all. But here is removal of base, here is addition of base. This is the only concept. And yes, in this part, one more very very important thing. If this could be MCQ, which one remain undigested food? So cellulose is always undigested till this part. And yet we really don't eat cellulose. If it was possible for us to eat cellulose, we would have eaten the food herbivorous plant, and not plant but herbivorous animal do it, like sheep, goat. They do it green type of food that contain a lot of cellulose, and in their stomach there is one enzyme that helps in digestion. We cannot really eat that. So till here it's like completely. And yes, wait, peptidase is used somewhere else. Here the enzyme would be nucleotidase, which help breakdown of nucleotide into nucleoside, and peptidase that convert into peptides. Peptidase convert into